Hello, I'm Dr. Gor Sarkisian. This is BIO425, Cell Biology. Today's lesson is on Chapter 5 from Campbell Biology, entitled The Structure and Function of Large Biological Molecules. Now, large biological molecules are involved in all of the aspects of the cell's life cycle, which is storage of genetic information, transcription of genes, cellular communication, cell replication, and every other cell-related function is performed by a group of biological macromolecules. So many molecules in a cell are macromolecules. It is not to say that there aren't any small molecules there. There are. Water, for example, is small and quite abundant. But when we talk about macromolecules and we talk about large sizes, how do we talk about it? Well, we have introduced a measuring unit called Daltons. An average macromolecule in a cell are in the order of 100,000 Daltons. 1 Dalton equals to 1.66 times 10 to the minus 27 kilograms. In perspective, a water molecule is 18 kilodaltons, and an average protein size is 100 kilodaltons. So in comparison, you can see that macromolecules are actually huge. So in this chapter, we will review how these macromolecules are built. We will examine the structure and function of all 4 classes of large biomolecules. These classes of biomolecules include carbohydrates, lipids, proteins, and nucleic acids. So what are macromolecules? Macromolecules are basically polymers built from their corresponding monomers. And to build a polymer from monomers requires energy. And to aid in such reactions, cells utilize a number of enzymes that enable polymerization of monomer units into macromolecules. So 2 monomers come together to form a chain of a polymer in a dehydration reaction. What exactly is dehydration reaction? It's basically a release of a water molecule when a covalent bond between monomers is made. As you can see on image A, we have a short polymer and we have an unlinked monomer. And as you can see, we have a hydrogen at the end of the short polymer, and we have an OH group on the monomer. So when these 2 systems undergo chemical reaction, a covalent bond is formed between a monomer number 3 on the short polymer and the subsequent monomer number 4 that is linked with that initial polymer. As a byproduct, we have a water molecule. Now, formation of macromolecules is not the only chemical reaction that occurs with polymer monomer systems. The opposite is also true, in which a polymer is being dissociated into its corresponding monomers. Such reaction is called hydrolysis, which comes from Greek hydro meaning water, lysis meaning break. So hydrolysis is exactly the opposite of dehydration reaction, in which a water molecule is absorbed and in so doing a covalent bond between the individual constituents of a macromolecule are broken, and an OH group is added to one of the former constituents of a macromolecule, and a hydrogen is added to its former covalently bound partner. So now let's consider an applicable example of hydrolysis. For example, in a digestive system. In a digestive system, an organic polymer from food enters the digestive system, but as such it is way too large to enter the cells. So within the digestive tract, a number of different enzymes attack these polymers and speed up the hydrolysis and basically produce a number of monomers, which are then absorbed into the bloodstream and are distributed throughout the whole body to all the cells. Cells then uptake these monomer building blocks, and in the cells, these monomers undergo dehydration reactions, and therefore the cells reassemble these monomers into different polymer macromolecule proteins that the cell needs. Now let's discuss the first class of our biological macromolecules, which are carbohydrates. Carbohydrates are both fuel and building material for cells. So carbohydrates, or sugars, are basically molecules that are multiples of CH2O, one of the most basic units, which is glucose. It's C6H12O6. As you can see, it's a multiple of CH2O. And glucose is the most common monosaccharide. Monosaccharide meaning a single molecule of sugar. And it is basically, it's essential for the chemistry of life. There are two main trademarks of sugars. It is a carboxyl group and a hydroxyl group. All sugars have these two groups in them. The monosaccharides, such as glucose, are a major part of nutrition for cells. In the process of cellular respiration, the cells extract the energy from glucose molecules by breaking them down. So besides being a major source of energy for the cells, the carbon skeleton of a sugar acts as a raw material for synthesis of other types of small organic molecules, such as amino acids and fatty acids. Now, the sugar molecules can be immediately used, but if they're not, they can generally be incorporated as disaccharides or polysaccharides, which are polymers of monomeric sugars. Now, here are different examples of sugars, and they're categorized in several different groups. First of all, a sugar could be a triose, pentose, hexose, which basically depicts 3, 5, or 6 carbons in the chain. They can also be in aldose or ketose conformations. Basically, aldose means the carboxyl group is at the end of the carbon skeleton at the top, as you can see denoted with the orange box. And the ketose is a carboxyl, the molecule with carboxyl group within the carbon skeleton. Now, look at the first pair, for example. These are basically, from the chemical formula standpoint, the same molecules, C3H6O3. However, the arrangement of individual constituents makes the chemical and physiological properties of these sugars different. Now, same thing applies with to pentose and hexose sugars. Another interesting and important variable here is depicted in a purple box. The differences between glucose and galactose. Now, these two molecules are enantiomers, as highlighted with the purple box. The only difference is that OH and hydrogen are switched in switched in places. This seems to be not a big deal, but when we are viewing a biological system in a three-dimensional system, this plays a big role. Now, these sugars are drawn in a linear form for convenience, but in reality, in aqueous solution, 5 and 6 carbon sugars are actually rings, because ring structures are the most stable in physiological systems. And it might ring a bell, a ribose. A ribose that we know of is a ring structure that is a part of the backbone of, that's right, an RNA. So here is what happens with the linear and ring structure of sugars. Now we have a carboxyl group at the top and hydroxyl group. And we have basically a covalent bond being made between the oxygen and carbon 1. So basically it's the oxygen attached to carbon 5 and carbon 1 itself. We are not losing anything. All of the constituents are there. Hydrogen is there, the oxygen is there. So the molecular formula is not changed. And in the end we have a ring structure, which is a more favorable conformation in the physiological systems. But as you can see from double arrows, these reactions could be reversible. Review question 1. Please describe what is a hydrolysis reaction. Question number 2. What is the basic theme of sugar? question number 3. What are the 2 essential functional groups that make up sugar? So as we have previously noted, a monosaccharide that is not immediately used can be stored in a disaccharide form. So, how is a disaccharide synthesized? For example, in image A, you can see a dehydration reaction in the synthesis of maltose. Maltose is basically a joining of 2 glucose molecules. So 2 glucose molecules are joined together at carbon 1 and 4 by forming a glycosidic linkage. And glycosidic linkage is basically carbon-oxygen-carbon linkage. Okay? And we have a functional maltose molecule. And if the glucose monomers are joined in different ways, it would result in a different disaccharide. Now let's look at image B, which is also the dehydration reaction in the synthesis of sucrose. And sucrose is a dehydration reaction between glucose and fructose. Now, this reaction occurs between carbon 1 of glucose and carbon 2 of fructose, and it is again a similar, it's the same bond as in maltose, which is a glycosidic linkage. Now, in plants, obviously the disaccharides are sucrose. In milk, we all know and have heard of, it's lactose. Lactose is basically glucose linked with galactose. And these obviously must be broken down to monosaccharides to be used for energy. And we all have probably heard of lactose intolerance. And lactose intolerance, it's basically absence of an enzyme called lactase. That's the enzyme that breaks down lactose into monosaccharides. So in those people who have lactose intolerance, the sugar is actually broken down by intestinal bacteria instead of the lactase. And that in turn causes formation of gas and subsequent typical cramping. So obviously, as you may have guessed, a polysaccharide would be a macromolecule polymer with few hundreds to few thousands of monosaccharides within its chain. These polysaccharides may serve as storage of energy, and they can be hydrolyzed as needed to provide sugar for cells. Polysaccharides may also be used as building material for structure that protects the cell or the whole organism. In case of insects, for example, the chitin is a polysaccharide. And but altogether, the architecture and function of polysaccharide is determined by the sugar monomer that is used to build the polysaccharide, and the position of the glycosidic linkage between the individual monosaccharide constituents. So here are several examples of polysaccharides. In picture A, we have starch, which is stored in plant cells. It is a polymer of glucose, and it is composed of amylose, which is an unbranched chain and amylopectin, which is somewhat branched polysaccharide. Next, we have glycogen, which is found in our muscle cells. Glycogen is highly branched polysaccharide, and it can be used for as nourishment for muscles. Yet another example is cellulose in plants. And these are polysaccharide structures that surround the cell wall and give the plant cells, their structure. So these are basically polymers of unbranched cellulose molecules. These molecules form hydrogen bonds between parallel cellulose molecules that hold them together in a bundle. Rather, these bundles are called microfibrils that bunch about 80 cellulose molecules together to create this matrix that gives structure and rigidity to plant cells. Now, we have stated already that the architecture and function of polysaccharides are determined by its sugar monomer and by the position of glycosidic linkage. So here's a visual example of alpha glucose and beta glucose. As you can see, the only difference is the OH group being on the same side with the adjacent OH or on the opposite sides. And these two conformations can coexist in certain equilibrium, and they can go back and forth between these two conformations. And as you can imagine that depending on which conformation the sugar is in, the glycosidic linkage with that C1 will result in slightly different conformation of a polysaccharide. So here we have examples of linkage of alpha glucose and beta glucose. As you remember from the previous slide, the only difference there is the conformation and the relative position of OH groups. Now, in case of starch, which is figure B, we have all the hydroxyl groups, OHs, aligned on one side of the polysaccharide. On image C, we have cellulose, which is again 1-4 linkage of glucose, but the glucose in this case is in a beta conformation. And you can see that the OH groups are alternating above and below the plane of the polysaccharide. Now let's steer away from carbohydrates and talk about fats. Fats are also essential for life. These are hydrophobic molecules. Fat is constructed from two kinds of smaller molecules, which are glycerol and fatty acids. And on this image, the glycerol is depicted with the gray background, and fatty acid is depicted with an orange background. Now, a classical dehydration reaction occurs between the carboxyl group and hydroxyl group on the glycerol to form water and form a covalent linkage between glycerol and fatty acid. Review question 1. What is a glycosidic linkage? Review question 2. What are the polysaccharides made from? Alpha glucose and beta glucose. Now, to make fat, we need 3 fatty acids being joined together to a single glycerol by ester linkage. Ester linkage is denoted on this picture in a red square. Okay? And again, these bonds are formed through a dehydration reaction between the hydroxyl group and the carboxyl group. And another word for fat molecule that we all heard of are triglycerides. And triglycerides, because this is the general form of fat. One molecule of glycerol with 3 molecules of fatty acids attached to them. Now, fats can be in 2 general forms: saturated fats and unsaturated fats. Basically, saturated fats include butter, which at room temperature is semi-solid. Okay? It is basically a triglyceride with a linear zigzag of carbon-hydrogen bonds without any disruption in their pattern. As you can see on a space-filled model, the saturated fatty acid is composed of an oxygen, carbon, and hydrogen in a straight line. Now, unsaturated fats are our everyday olive oil, which is liquid under normal conditions, and the only difference is that it has a kink in it. It has one double bond carbon-carbon covalent bond in at least in one of the branches of the fatty acid. Okay, in this particular conformation, as you can see, it's in cis double bond. Cis being the conformation goes towards the same side of the molecule. Now, we all know that some fats are bad, and for example, the saturated fats can result in atherosclerosis, which is the development of a plaque on walls of blood vessels, which can restrict blood flow. But on the other end, we can have a trans conformation of unsaturated fatty acids, and we have also heard of trans fats being bad for you. Well, we will talk about trans fats later in the course. Now, we can't talk about fats without talking about phospholipids. Phospholipids are essential for life. These are the molecules that delineate our cells, that protect our cells, that allow signals selectively to reach the inside of our cells, and they form a very, very nice barrier. And the overall structure of a phospholipid is as follows. It has a choline head, phosphate, glycerol, obviously, and 2 fatty acids, and one of them is unsaturated. Now, having such peculiar shape gives a phospholipid very interesting properties, in which on one end it's hydrophilic. It likes to be attracted to water. On the other end, it's hydrophobic. The tail, the fatty acid tail, is hydrophobic. So if we have a mixture, a solution of phospholipids, and we pour that solution over a body of water, into a glass of water, we will have an orderly arrangement of phospholipids facing the water with their hydrophilic head and having the hydrophobic tail sticking out. That would create a phospholipid monolayer. Now we can add another layer of phospholipids to that, and that will stack up with hydrophobic tails being drawn towards each other. And as you can see on figure D, that's exactly what we have there. We have a phospholipid bilayer. And we can have water on each side, and that will keep this bilayer in a stable condition, and it will remain intact. Review question 1. What is an ester linkage? Review question 2. What makes an unsaturated fat? Now, the other form of lipids are steroids. And one prime example of steroids is cholesterol. Now, cholesterol is important, it's essential. It acts as basis for many different hormones, and it's an integral part of the cell membrane. But of course, a lot of cholesterol can clog the arteries and result in very undesirable outcomes. But it is true that cholesterol can be modified to create cortisol, progesterone, and a number of other sex steroids. And finally, let's talk about proteins. Proteins are quite diverse in size, in shape, in function. The word protein comes from, guess what? Greek word proteos, which means prime or first. The proteins account for more than 50% of dry mass of a living cell. Some proteins can act in speeding up chemical reactions. Others play a role in defense, storage, transport, cell-to-cell communication, movement, structural support. We will talk in much more in depth about proteins later in the course. And probably the amino acids are not a direct part of this course, but please advise if you'd like to dive deeper into amino acid biochemistry, and I would be more than happy to do so. Review question 1. Please describe the hydrophilic head of a phospholipid. Now let's talk about some functions of proteins. Proteins could be enzymes. Enzymes are basically vehicles, in this particular case, to selectively accelerate chemical reactions. A prime example is a digestive enzyme that can hydrolyze the bonds in food molecules to break them down to monomers and make them bioavailable. Proteins can act as defense mechanisms, protecting the system against pathogenic invaders such as bacteria. Antibodies, for example, are a very, very efficient tool to protect us from the immune system. Antibodies are basically tags that attach to a foreign body, and they either trigger immune cells to directly eat these foreign bodies, or they can trigger a more specific immune response, that is directed to specifically that particular bacterium or virus or any other pathogen. Another function of proteins is storage of amino acids. An example is casein, a major protein in milk, which provides amino acid to babies. Another example is of albumin, which is basically egg white, and it is also a major source of amino acids for developing embryos. Another function of proteins is transport. If a protein is embedded in a cell membrane, it can act as a transport machinery to selectively allow particles in and out of the cell. Another function is hormonal proteins, which coordinate and orchestrate the organism's activity. An example is release of insulin in response to high blood sugar. Insulin, in turn, reduces the amount of sugar in the blood, bringing it down to its normal levels. Another function of proteins is receptors. Receptors are basically proteins that respond to chemical stimuli. Receptors can be built into the membrane, for example, of nerve cells, and they can detect molecular signals that are released from other neurons, and they can respond accordingly. Now the contractile and motor proteins, muscles, which are involved in movement. The motor proteins are responsible for undulation of cilia and flagella. Actin and myosin proteins are responsible for contraction of muscles. Actin and myosin are basically the constituents of a muscle fiber. And of course we have structural proteins that play a big role in keeping these shapes and conformations intact. An example of such protein would be keratin, protein in feathers in hair. The spider's silk is also such a protein. Collagen in an extracellular matrix are all parts of the support machinery of the supporting protein structures. So here are three different ways that we display proteins to make sense of them and to make the information useful. The space field model is basically an image that shows the atoms of the protein, obviously except for hydrogens. And this emphasizes the globular shape. And there are certain color codes. In this particular case, gray for carbon, red for oxygen, blue for nitrogen, and yellow for sulfur. And this holds true, but of course there are other molecules that are involved that are probably less prevalent. Next, we have the ribbon model. This only shows the backbone, and it emphasizes how the protein is coiled, or how it's shaped into its 3D shape. It can show the unimportant bridges like disulfate bridges depicted in yellow, We will talk about this later. These are some of the most essential bridges in a protein. Then we have the wireframe model. In this particular case, we have a superimposition of the ribbon model, right? And we have the chain with side chains, the R groups, extending from its the main backbone structure. All of these have something to show, and in different applications, one or the other comes in very handy. Review question 1. Please describe the process of formation of monomers from polymers. Review question 2. What is a catalyst and how does it operate? So all in all, the proteins can be divided into several structures. Obviously, the primary structure, which is nothing more than a sequence of amino acids, which are later combined into a secondary structure, and there are two main ones, the alpha helix and beta sheets. These can be packaged together into a tertiary structure, which is a three-dimensional shape of a protein which is stabilized in its environment. And the fourth level, the quaternary structure, is the association of several different proteins that actually form a functional unit. Mutations in proteins can be very detrimental. An example of a single amino acid mutation is in sickle cell anemia. In sickle cell anemia, basically a glutamic acid is mutated by valine. And this basically forms a hemoglobin that is sticky. Hemoglobin should not be sticky. It should be free to interact with oxygen, basically to have free carrying capacity for oxygen. Now in a sickle cell anemia, this is not the case, and this sticky form of the hemoglobin also results in abnormalities in actual shape of the red blood cell in a sickle cell anemia disorder. Well, thank you very much. Please note that there are at least 10 sporadically distributed throughout the presentation questions, which are going to be counted towards your participation and as an assignment for the week. So please make sure you find all of them. Some of them are written on the slide and some of them are spoken. So if you find at least 10, you're in good shape. If you find anything beyond 10, it's extra credit. It's your call. Once again, thanks and have a good day. Review question 1. What is quaternary protein structure?